Hi everyone, my name is Hannah Zang and I am a junior this year double majoring in biology and psychology. And today I'm going to be talking about the research that I did at the University of Arkansas in Fayetteville through the Embry Summer Fellowship Research Program. So this was a 10-week program and my project largely consisted of functionally characterizing a neuropeptide in Nematostella victinicis, otherwise known as the starlet sea anemone. So to explain how I did this, I used a CRISPR-Cas9 mediated knockout approach, and I'm going to explain CRISPR-Cas9 and how it works through simple drawings. So when we do CRISPR-Cas9 microinjections, we are actually injecting what we call a CRISPR cocktail. So a CRISPR cocktail has three components. We have the guide RNA, Cas9, an endonuclease, and a dye. So basically, the guide RNAs are what identifies the gene of interest. And in this case, I was interested in identifying the gene that was responsible for producing the neuropeptide RPamide. So they act as red flags or traffic cones and basically says this is the spot that we are interested in studying. Next, we have Cas9, an endonuclease. And the way I like to think of this is molecular scissors or molecular knives. They'll basically go to the region where the guide RNA is pointed out and then cut that region. So at this point, you are basically getting rid of the gene of interest. And when you do microinjections, you also want to include a dye. So that is the last final component of the CRISPR cocktail. And then you are injecting this CRISPR cocktail into an embryo at the single cell stage. So the reason why you want to inject at the single cell stage is to prevent mosaicism. Mosaicism is when the egg starts to divide and then you inject the CRISPR cocktail. Likely you wouldn't be able to incorporate the CRISPR-Cas9 cocktail into all of the cells that have divided. So that's why you want to inject at the single cell stage. So after you have injected all three components into the embryo, you wait a day. And after a day, you should be able to identify if they are actively dividing. This is because a single cell stage is what you would expect. It is round, it is egg-like. And then after a day, it will be bumpy, kind of like crystallized at the surface, which represents all the tiny little cells that have divided. And then you can look at it under a fluorescent microscope and it should light up because of the dye. So hopefully you have injected the CRISPR-Cas9 cocktail successfully into the eggs and that they are also showing the dye, meaning that they have been injected. So after this, you let them grow and then you do DNA extraction and polymerase chain reactions to amplify the DNA. And then you can visualize the DNA using gel electrophoresis. So the point of this is to knock out the gene. We have the guide RNAs and the Cas9, and they are functioning and getting rid of that gene. And by doing this, you are perturbing the ability of the sea anemone producing that gene or protein that will eventually produce. So I created an F0 population. This is the first population that lacks the protein of interest or RPamide. And then I grew them up and attempted to phenotypically characterize the sea anemones, identifying possible phenotypes such as life cycle transitions, since sea anemones undergo metamorphosis, or possibly muscle contraction. And the goal of my project was to publish a paper. So I published a paper in Frontiers of Endocrinology with my mentor, Dr. Nagayasu Nakanishi. Feel free to check that out. And I also got to present the work at two different conferences in the Central Arkansas Undergraduate Summer Research Conference in Little Rock and at the Society for Integrative Comparative Biology Conference in Austin, Texas. So hopefully that explains how CRISPR-Cas9 microinjections work. And CRISPR-Cas9 can be used beyond seed enemies. It actually has a very important implication for gene editing, and eventually CRISPR-Cas9 may be able to eradicate human diseases using gene editing technology. So hopefully you can understand how CRISPR-Cas9 works, and specifically with microinjections. And if you have any questions, feel free to email me at hannah.zang@lion.edu, and I would be happy to answer any questions that you have.